Aloha. Welcome to part 2 of lecture 10. In part 1, we talked about basic brain anatomy, development of the very early development of the central nervous system, and then we went over the development of the midbrain and hindbrain primarily. Cervical flexures, primary brain vesicles, secondary brain vesicles, all that kind of stuff. And then we ended with the early development of the midbrain. So that leaves us with the forebrain. Let's tackle this guy. He is a beast. This is the most complicated biological structure known. So the rostral neuropore closes. As it closes, you start to see these two lateral outpouching. These guys are called the optic vesicles, right? The optic vesicles, as the name suggests, are destined to become the optic nerve and the retina of the eye. Not much later, on the top and towards the front, back it up. On the top and towards the front, you're going to see two more outpouchings form on top of these. So there's this outpouching here, and there's going to be another outpouching on top of that and towards the front, right? That secondary outpouching on the top and to the front is called the telencephalic vesicle. The telencephalic vesicles are eventually going to become these guys. Now, later in the book, they don't call them the telencephalic vesicles anymore. They call them the cerebral vesicles, and I'm fine with that. But just so that you're aware, the telencephalic vesicles and the cerebral vesicles are the same structure. They are the same thing. I can't remember which one shows up on the quiz. I believe one of them does. But it's going to be really important for you to know that the first lateral outpouchings are the optic vesicles, not the telencephalic vesicles. The optic vesicles come first, and then the telencephalic vesicles are a secondary outpouching from the optic vesicles. Hopefully I made that very, very clear. So yeah. The cerebral hemispheres, the cavities of these vesicles are destined to become the lateral ventricles. The front part of the brain is called the telencephalon, and the back part of the forebrain is the diencephalon. So we've got, the telencephalon is the front part of the forebrain, the diencephalon is the back part of the forebrain. They're still the forebrain, but different sections of it. The cavities of both are going to contribute to the formation of the third ventricle. And anyway, so let's tackle the diencephalon first. 93, here we go. The diencephalon. So this is a 7-week brain over here, and this is an 8-week brain down here, okay? So in the 7-week brain, you start to see these three swellings appear. These three swellings are the thalamus, and then above the thalamus is the epithalamus, and below the thalamus is the hypothalamus. They make a lot of sense, those names. Now, the third ventricle comes right through these guys. It comes right on through all of them. On the floor of the thalamus, you start to see. Make sure I lost my place here. Yeah, okay, I'm in the right spot. So yeah, on the floor of the thalamus, you start to see these this swelling occur, this swelling of cells. It's a proliferation of neural cells, and this is the mammillary body, right? So the mammillary bodies are nuclei that play a role in memory recall, as we mentioned before, which if you remembered, your mammillary bodies are working. But over the next week, what's going to happen is the thalamus is going to way outgrow the epithalamus and the hypothalamus. Right here he is down here, way bigger than his brothers. So here's the epithalamus, here's the hypothalamus. And while this happens, right out of the roof of the diencephalon, you're going to start to have this little nipple forming. And this little nipple is going to differentiate into glandular tissue. And this is going to become the pineal gland, which regulates melatonin in the brain, right? It controls your sleep-wake cycle. Another thing you'll notice already forming during week 7 is this little diverticulum down here. This little diverticulum is called the infundibulum. This is coming out of the hypothalamus. He starts forming actually as soon as the embryo begins to fold, and this is going to ultimately become the neurohypophysis, otherwise known as the posterior lobe of the pituitary gland. Now, the book harps on the formation of the pituitary gland, and that's appropriate because this guy controls so much of the hormone symphony in the body. It's just crazy how much it does. Not to mention the fact that it forms in a very unique way that people like to put on quizzes and tests and things like that. So we're going to spend a little bit of time on the formation of the hypothalamus, or of the pituitary gland. So anyway, as the embryo folds, the infundibulum forms. So does this diverticulum in the stomodium, and this is called the hypophyseal diverticulum. Now, this is formed. It's made out of surface ectoderm in the mouth. This is surface ectoderm. This is neuroectoderm. That's going to be important later. We're going to reiterate it again and again and again. But the diverticulum stems from the surface ectoderm of the mouth, the stomodium. And as the diverticulum deepens, as this thing caves in further and further, it's going to eventually come in contact with this diverticulum. Come on, my mammillary bodies are failing me. The infundibulum, thank you. So yeah, the infundibulum and the hypophyseal diverticulum are going to come in contact with each other eventually. Now, the infundibulum, as I mentioned, is formed from neuroectoderm. The hypophyseal diverticulum is formed from surface ectoderm in the mouth. Neuroectoderm from the floor of the brain, surface ectoderm from the mouth. Okay, now, as this infundibulum contacts this hypophyseal diverticulum, this guy is going to turn into a pouch and it's going to start to be pinched off. As the stock of the hypophyseal diverticulum gets pinched off, it's going to regress. Now, there are these little leftover bits. For purposes of the class, we are just not going to worry about them. They don't do anything that's important to us, okay? But there are little bits of tissue left over from this regression. What does matter, though, is that once the hypophysis is disconnected, it differentiates into glandular tissue. This lumen that's left over, this hollow spot in the middle, when it gets pinched off, is going to break up into vesicles, and these vesicles contain colloid material. Now, nobody knows for sure what it does, but some experts believe that these vesicles serve a similar function to the thyroid. They kind of assist the thyroid in modulating the metabolism. So the infundibulum also develops into a bulbous collection of nerve cells. So this guy starts to turn into this nice little bulb of nerve cells, and this is going to eventually become the posterior part of the pituitary gland. So you have the anterior pituitary gland and the posterior pituitary gland. Of course, the skull then closes in around it, encasing it in bone. And it's going to be important to remember that the anterior pituitary is formed from surface ectoderm of the mouth, And the posterior pituitary gland is formed from neuroectoderm cells coming from the floor of the brain. I just want to reemphasize that one more time. Okay? Okay. Now, the telencephalon is a little bit easier to talk about. It basically just gets bigger. You start with these outpouchings, the cerebral vesicles, which remember I said are the same thing as the telencephalic vesicles, which have these little cavities in them. And these little cavities, as these guys get bigger and grow, these little cavities are going to get bigger and grow with it. And they're going to follow the growth of the hemispheres. But anyway, so these cerebral vesicles are going to get bigger, and they're going to get bigger very, very fast. So as they do that, the ventricles just follow it. Let's see. So anyway, the brain's getting bigger up here. This guy's getting bigger and bigger and bigger. But it's also developing nuclei, commissures, and things of that nature. So you have this really big cluster of nerve cells and fibers called the striatum that forms the floor of the brain. Right? The striatum is down here. And then you also have the corpus callosum, you have the hippocampal commissure, you have the anterior commissure. You got all these structures on the bottom of the cerebral hemispheres that grow kind of slow. Meanwhile, the hemispheres themselves are growing really, really, really fast. This is where the C shape of the brain comes from. As this area outgrows this area much faster, this area stays small, this area stays big. You can kind of see what's going to happen. It's going to form this nice big C shape of the brain. So it might be worth remembering for a quiz in the near future that the C shape of the hemispheres of the brain, the C shape of the hemispheres results in structures on the bottom of the brain growing relatively slow compared to the rest of the hemispheres. Hopefully you got that. Now again, this is going to show how these ventricles form. So you have these fluid-filled spaces, and as the hemispheres grow in this C-shaped pattern, the ventricles, the fluid-filled spaces, they just follow right along and grow along with the brain. So it might be worth remembering that the C-shaped ventricles, you end up with C-shaped ventricles because the existing spaces follow the growth of these cerebral hemispheres. That might be something that might also appear on a quiz in the near future. So also before long, these hemispheres outgrow the diencephalon and basically every other structure in the brain until they meet in the front and at the top. And as they meet in the front and the top, they squash up against each other, and that forms this nice flat surface that you see in the center of the brain. If you've ever looked at a brain sliced in half, the center surface of the brain is a real nice flat surface, and that's because they overgrow everything else and they butt up against each other in the middle. So there's nowhere for them to go except to flatten out against each other. Now during week 7, I'm jumping ahead of myself here. During week 7, the telencephalic choroid plexus forms, and the mechanism of how the choroid plexus in the lateral ventricles forms is really similar to the mechanism in the fourth ventricle. You have thin spots in the pia mater, and at those thin points, the pia mater proliferates and invaginates into the cavity, since that's the only place for it to go. And then once those cells invaginate into the cavity, they differentiate into these choroid plexus cells that produce cerebral spinal fluid for the central nervous system. And this occurs in the medial dorsal surface of the ventricles. So towards the middle, towards the top of the brain. The middle top of the ventricles. Eventually, yeah, so this is just showing again, the ventricle follows the brain. This part's growing slower, this part's growing faster, so it starts to grow around and the ventricle follows it. But eventually what's going to happen is we're going to run out of space, right? You're going to hit the bottom of the skull. So as this thing hits the bottom of the skull, it has nowhere else to go. So it just wraps around towards the front, right? Hits the bottom, wraps around towards the front. And as it wraps around towards the front, which forms this temporal lobe down here, forms the temporal lobe, then the ventricle follows. And that forms the temporal horn of the lateral ventricle. Then it still has not enough space. So this occipital region starts to push backwards because there's a little bit of space back here. So this occipital region of the brain starts to push backwards into this little space in the brain, which causes this occipital horn of the lateral ventricle to form. So you see, this is the same story all the way around. The ventricle just follows the growth of the brain as it wraps around and then wraps around to the front and pushes into the back. You form the different horns of the lateral ventricles. So eventually we're going to run out of space, right? We've filled up the back, we've filled up the front, we've wrapped around in a circle, we've filled up every nook and cranny that there is inside this skull. The problem is we're not done developing. We still need more surface area for cortex. We have many, many, many neuron cells, many, many neuron cell bodies that still need to be proliferated and developed. And, the head can only get so big and still go through a birth canal. So what's the solution? Well, the brain starts to form these gyri and sulci. So you have one gyrus and multiple gyri, you have one sulcus and multiple sulci, right? So these gyri and sulci begin to form, which are basically just hills and valleys in the brain, and this increases the surface area, which allows a greater amount of surface area to house the gray matter, to house all the cell bodies, all the cortical cell bodies, right? So you notice down here, this is a 22-week brain, and this 22-week brain has a pretty smooth surface. It looks pretty smooth. Now, let's compare that to a 40-week brain. Oh my gosh, quite a bit of difference, right? Here's a 40-week brain. Look at all the hills and valleys and gyri and sulci and all that stuff. And again, what this does is it creates more surface area for brain cells to exist, more surface area for blood flow to exist. It serves a lot of functions, but the whole point behind these gyri and sulci is to create more surface area on the brain. Because again, we've used up every tiny little nook and cranny in the skull that we can to fit this very huge brain. And this allows for adequate cortex formation without a grotesquely enormous head, right? Now, next question I have for you is this 40-week brain fully developed? And the answer to that question is absolutely not even close. So my next question for you is, when will this brain be fully developed? When will the final connections be made? When will the last of the cortical cells be? How long does it take for the brain to develop all the way? So the answer is it depends, right? And largely it depends on your gender. So for women, this happens earlier in life. Women, it's about age 19. By the time they're age 19, those last connections are made and the brain is fully formed. For men, it happens much later, at about age 25. You ever notice how women's car insurance drops significantly at the age of 19 and the cost of a man's car insurance drops significantly at the age of 25? Well, this is why. The last connections of the brain don't happen in men until they're about 25 years of age. So guys, I'm really sorry, but most of you in this room do not have a fully formed brain, while all of you women, your brain's good to go. I'm sorry, guys, that's just the way it is. At any rate, we have now finished our discussion of the brain. That is going to be it for part 2 of lecture 10. Next is going to be lecture 11, where we're going to start discussing the spinal cord and the peripheral nervous system. Like I always tell you at the end of every lecture, if you have any questions or need clarification on any of this material, please write your questions down, bring them with you to the Zoom meeting, and we can give you any clarification that you need. If you're not able to make it to the Zoom meeting, ask one of your friends to bring the question up for you. It really is my favorite part of the class when I get to interact with you guys. So that'll do it for now. We'll see you guys at the next lecture. Good luck at the quiz. I hope to see you at the Zoom meeting, and aloha.